Hello and welcome to another episode of Momento Malum today. The mung bean is an important pulse crop in India. The overall improvement of the species currently depends on the amount of genetic variability present in the base population. The genetic diversity is an important factor and a prerequisite in any hybridization program. The mixing of genetically divergent parents in breeding projects serve as a means of creating a wide array of genetic combinations, each holding the potential to become a brand new cultivar. Unlike most other genetic analysis, the researchers behind this paper did not use genetic markers, but instead used a statistical method called Mahalanobis D2 statistic, a strategy that categorizes cultivars based on a set number of traits and their relationship based on said traits. The traits analyzed would be number of days to first flower, plant height, number of branches per plant, number of flower clusters per plant, number of pods per plant, number of pods per cluster, pod length in centimeters, pod weight in grams, number of seeds per pod, and how much 100 seeds would weigh in grams, as well as seed yield per plant. The mung bean lines were analyzed using this methodology, and here are the results. The results found that the 30 lines were grouped into 6 genetically distinct groups, seen on screen. The last 2 groups consisted of 1 line each. The data also indicate that there is significant variability existing among the genotypes, as observed from each individual group. Cluster 2 consists of specimens from different regions. This indicates that there is no correlation between how the lines group under this method and the geographic distribution of these genotypes. The estimated genetic distance was lowest between groups 1 and 2, and the highest differentiation between different groups was between groups 1 and 5. This means that crossbreeding between groups 1 and 5 will yield far more divergent genetic lines, considering the fact that the greatest variability in terms of traits analyzed was yield. This may further increase yields over time, due to the amount of variability that will be created through the crossbreeding of these 2 genetically divergent lines. And that about covers everything. Thank you for watching. If you want to donate to my channel, the links are below. If you want to buy my book, you can also find that link in the description box below.